Mike asked me to tell you a little bit about how I got into this area, and I definitely intend to do that. But one thing that I wanted to point out about what he said about my training is that it was a very conventional, traditional Western medical training, and really didn't involve anything to do with herbal remedies, like I'm sure many other physicians of my generation. And so what I hope to do by the end of this hour is give you some new information that will hopefully be helpful to you as healthcare providers and also as individual consumers of herbal remedies, because you may be surprised at how mainstream some of these remedies have become and how they've become additives to very common things like multivitamins and drinks, sport drinks, shakes, things like that that are supposed to be healthy for us. So on this first slide, I've listed the topics that I plan to cover today. And the first is, how did I get here, to answer Mike's question. And then we'll cover some background about CAMs in general, and then specific issues that have to do with herbal remedies, safety, standardization, regulation, and then the promise of herbal remedies. And finally, I'd like to discuss some specific progress that we've been making in Silymarin research here in the Department of Laboratory Medicine. So hepatitis C virus is the virus that we study in our laboratory, and it is the most common chronic bloodborne viral infection in the US. It infects millions of Americans and people worldwide, and it is a significant cause of death and a leading indication for liver transplantation in the United States. And currently there's no vaccine for this infection. So shown in this slide is a description of the natural history of hepatitis C virus infection. And what you can see is that of people who are acutely infected, we believe that the vast majority of them develop chronic infection. And that of those people, most of them develop active hepatitis in their livers. And then some subset of them develop a scarring response or fibrosis in their livers as a response to this chronic inflammation, and that over time, as you can see over a period of decades, that some of them develop cirrhosis, complications of cirrhosis, liver cancer, and death. And one of the things that I think is important to point out here is that the transmission of hepatitis C virus has changed significantly, and that really had a lot to do with the advent of very sensitive molecular diagnostic techniques for detecting hepatitis C virus in blood and blood products, which was a primary mechanism by which hepatitis C virus was transmitted. And so currently, the most common method by which hepatitis C virus is transmitted is through injection drug use and use of shared needles. And unfortunately, why do I keep doing that?
I don't know what that is.
Prior to the advent of this testing, unfortunately, a large number of people were infected with this virus. And now it's been several decades since they've been infected. And so what's predicted is that in the coming decades, the people who were previously infected with hepatitis C virus are going to be developing these long-term problems associated with the infection, such as cirrhosis and the complications of cirrhosis, including the development of ascites and hepatic encephalopathy, variceal hemorrhage, et cetera, as well as the development of liver cancer or hepatocellular carcinoma and liver-related death. One thing that I want to point out about this is that this line looks sort of flat here for hepatocellular carcinoma, but it turns out that liver cancer is actually the most rapidly growing incidence of cancer in the United States, and that currently the average survival rate of a person diagnosed with hepatocellular carcinoma is less than 1 year. So this is a substantial medical problem. So there is a treatment for hepatitis C virus, and that's listed here. That's pegylated interferon alpha and ribavirin. The problem with this treatment is that it's difficult to take. It requires weekly injections, is lengthy. It usually requires either 6 to 12 months of treatment. It's costly, on the order of tens of thousands of dollars. And it's poorly effective for HCV genotype 1, which is the most common genotype to infect people in the United States. And for those people who are infected with genotype 1, the success rate of treatment is less than 50%. And I should note that it is possible to completely clear the virus and cure people of hepatitis C virus, although it doesn't necessarily eliminate the risk for potential future problems as a result of having had infection. In addition, there are significant side effects that make the treatment intolerable for some people, and there are contraindications to therapy so that some people are just simply not eligible to be treated. Well, it turns out that there is a new drug in development, which we hope will be available in a few years for people who are infected with hepatitis C, and that's called telaprevir, and it is a hepatitis C specific protease inhibitor. And the good news about this new drug is that it's very effective, and we expect that it will make a significant dent in terms of increasing the success rate of treatment. The downside of this new drug is that what it does is that it tends to make the virus mutate more rapidly. And as a result of that, we know that it will need to be used in combination with pegylated interferon alpha and ribavirin, so that people who are ineligible to take current therapy are unlikely to be eligible to take the new next step of therapy. And so apart from these options, there are actually no other FDA-approved options for people who are infected with hepatitis C, and there are no currently approved treatments for fibrosis progression. So maybe it's not such a surprise then that people have turned to Silymarin, which is actually an ancient herbal remedy that has been documented now to be used by up to 23% of patients with chronic hepatitis C in this country. And so it was this fact and this man whose unbridled enthusiasm overpowered me and convinced me that I should try to do some experiments looking at the immune mechanism of action of Silymarin in hepatitis C virus infection. And so I'll come back to these experiments towards the end of the talk. So right now what I'd like to do is spend some time giving you some background about complementary and alternative medicine, or CAM, used in the United States today, and the problems associated with herbal remedies, specifically with standardization and regulation of these products. So shown in this slide are the results of a survey that were conducted by the Centers for Disease Control in 2002. And they interviewed more than 31,000 people, adults, and asked them what are the most common, what are the CAM therapies that you use? And you can see here that number 1 and number 2 were prayer, but that after that, natural products was the top category. So when they asked these people more questions, one of them was, why are you taking these products? And these are the answers that are listed in sort of the order of popularity. And of course, they don't add up to 100, so people are able to choose more than one option. But the most common answer was, well, it was just interesting and they wanted to try it, which says something about, I guess, the open-mindedness of the American public, and may have to do with how easily these products have been accepted by Americans. The second choice was they thought that it would help with conventional therapy. The third was that conventional treatments won't help. The next was that a conventional practitioner had actually recommended that they use it. And then finally, that conventional treatment was too expensive. So there are two things that I wanted to point out about this list. The first is that people, the vast majority of people, or a large number of people, were actually going to take it, planning to take these herbals with their conventional drugs, okay? And subsequent surveys and other surveys have shown that most people who take these don't actually tell their providers that they're taking them. So then the other important point is that two of these options really imply that people were taking these herbal drugs instead of conventional drugs. And that may have been in situations where there were conventional treatments that would be more helpful to them than these herbals. We don't know that. Okay, so this slide is included just to show you that the herbal dietary supplement business is big business and is big money. So starting in the 1990s, when things were really taking off, you can see that there was an incredible growth period, and things have sort of stabilized now in the 2000s, but they've stabilized at this figure of about over $4 billion a year. So there's a lot of money that's being spent on these herbal supplements. And of course, the people who are manufacturing and distributing these products have a lot of money and use that money probably to influence legislation and other things that might be important for our health. So who takes these herbal remedies? Well, a lot of people in this room might actually take these because studies have shown that Caucasians, women, well-educated people, people of higher socioeconomic status, and those who live in this area are more likely to take herbal remedies. And in addition, people, as I stated to you earlier, with chronic medical conditions that perhaps are not adequately addressed by their traditional providers are more likely to, again, take these herbal remedies. So just for fun, I included this table to give you a flavor of the kinds of things that people are buying. And I particularly like this list because milk thistle was on this list. And I should say to you, however, that this list is generated yearly and looked at in various ways, and it's always changing. So the top 10 things in 2005 might not be the top 10 things this year. But the main thing that I wanted to point out to you is that herbal ingestion is very, very prevalent. 1 in 5 American adults use dietary supplements, and I have to qualify this because in this case, dietary supplements also include multivitamins, so it's not just herbal remedies, but 1 in 5 American adults used a dietary supplement in any given week or over the past 12 months in 2 separate surveys in 2002, and that about 15 million adults took prescription meds concurrently with herbal remedies or high-dose vitamins in 1997. So I would propose that this number has got to be significantly higher now than it was in 1997. So what about the safety of these herbal products? Well, they are perceived as safe because they're natural, right? But what I need to remind everyone is that herbs can contain potent bioactive substances. And I'd like to remind you that nearly a third of pharmaceutical drugs were originally derived from plants. And here is a partial list of the drugs that are commonly used to treat people for various ailments. And we'll talk a little bit more about Artemisinin in a couple slides. So, problems with the safety of herbal products. Well, as we alluded to earlier, taking herbal products can delay, replace, or compromise conventional treatment. In addition, there's variable potency for reasons that we'll discuss later in terms of the potency in different lots from different manufacturers and so forth. There's also difficulty in detecting adverse effects that occur over time. People can have allergic reactions to herbal products just like you can have an allergic reaction to food or to a drug. There are direct toxic effects. There can be contaminants or adulterants, and there can be interactions with other drugs and herbs. And so I'm going to give you a few examples of these last 3 categories in a minute. And I'll refer you to this really nice review by Peter Desmet in the New England Journal of Medicine. He also has a more recent one from 2004. And for those of you who are more interested in this topic, I actually have a list of references that were up by the front door, and you can get one on your way out if you would like. So this is the article published in the New England Journal of Medicine now in 2000, so it was a while ago. But this group in Belgium described the occurrence of rapidly progressive renal failure in a group of women who had gone to a weight loss clinic and been prescribed this Chinese herbal medicine for weight loss. And so they developed end-stage renal disease. And in addition to that, years later, it was found that this particular substance was also causing urothelial carcinoma. So there are a couple things here that I wanted to point out. And one is that the reason that this happened was because they presumed that there was some sort of manufacturing error that led to the inclusion of the wrong herbal product in the weight loss pills. That's number one. And then number two, there was a direct toxic effect of this particular contaminant or mistake in the herbal product. So there can be serious consequences of taking these things. The next thing that I wanted to point out is the presence of adulterants and contaminants. And this is data from Richard Koh, who was at the time at the California Department of Health Sciences. And what he and his colleagues did was they went to California's retail stores and they bought 260 medicines that were traditional Chinese medicines, that's what TCM is, and a type of them called Asian patent medicines. They read the labels and they also tested what was in the bottle. And so when they did that, they found that about 5% of the bottles that they looked at actually said there was they declared a drug that was also in that bottle. But 7% had drugs in them that weren't declared. And then between 10 and 15% had levels of lead, mercury, and arsenic. So overall, 32% had undeclared drugs or heavy metals. And just so that you don't think that this problem is limited to Chinese medicines and stores in California, there was a subsequent study that was published in 2004. And what these investigators did was they went to Boston area stores and they bought these Ayurvedic herbal medicine products and tested those and found that 20% of them contained unacceptable levels of heavy metals. So this is an example of heavy metals in drugs, and there can be lots of other things that are bad for you in these herbal remedies. And finally, I just wanted to mention drug interactions. So St. John's wort, for those of you who don't know, is a very commonly used herbal remedy for mild depression. And it turns out that this has been one of the best characterized in terms of having drug interactions with conventional drugs. And I should note that this is one of the few herbal remedies that actually has been shown to have some clinical efficacy in mild depression. So it's something that people are likely to use. And we as healthcare providers need to be asking patients whether or not they are taking herbs specifically, because they can interact with the medications that we're prescribing. So why have there been all these problems with these herbal dietary supplements? How can things be so messed up? Well, part of the reason is this legislation that was enacted in 1994. And what it did was that it categorized herbal products along with vitamins and minerals as a dietary supplement. And when you categorize something as a dietary supplement, that means it's a category of food and it's not a drug. And what the FDA does for drugs is it has very stringent requirements for drugs before they come to market, but they don't have those requirements for food. So in fact, there's no need for FDA approval before you go to market, okay? And in addition, the other provisions say that actually the manufacturer is on the honor system. They need to be sure that they are accurately labeling the product and telling you what's in it, that they're responsible for making sure that the product is safe, and they're responsible for making sure that any claims that they make about the product are actually true. And at the time, there was no minimum standard for how these products needed to be manufactured, no rules about the minimum or maximum serving sizes or amounts that people were supposed to take. There was absolutely no requirement to record, report, or investigate claims of injuries or illnesses associated with these products. And finally, these supplements were not registered with the FDA, which actually meant that if there was a problem with that product and the FDA wanted to contact the company, they didn't necessarily have the contact information to contact the company. And finally, the FDA had to prove that a market was unsafe before they could take it off the market or restrict its use. So this is a pretty shameful act. But luckily, there's been a little bit of improvement since then. It's taken quite a long time. But effective December of 2006, there's finally mandatory reporting required of serious adverse effects to the FDA that is required of manufacturers and people who package or distribute these products. So that's good news. And in fact, I should mention that there are ways that, there's a hotline and actually the website where consumers and healthcare providers can go and actually report adverse effects, which is a very important thing to do. But one of the things that the FDA realized was that with all these problems, people were actually contacting poison control centers, but nothing was really being reported to the FDA in comparison. So then there's the FDA final rule, which came out not too long ago in June 2007. And what the FDA had done was done some studies to try to figure out why there were all these problems and what could be done to try to resolve these issues so that all these problems wouldn't continue. And so what they decided was that if they set out a final rule on how these products should be manufactured, then most of these problems would ostensibly go away. And so the goal of this was to ensure consistency in the identity, purity, strength, and composition of these dietary supplements. And what they set were a staggered set of compliance deadlines according to the size of the company. So a really big company needed to comply by June 2008, and there are still some companies that don't need to be in compliance because they're very small until June of 2010. And so what they laid out, and this is a very, very hefty document that they laid out, were requirements for how the physical plant should be laid out, quality control procedures, and product ingredient testing. But they didn't really set any standards, and they didn't really say, well, they said, you, the company, have to decide what you want your product to be like, and it needs to be consistent, and you need to be able to identify that it is what you say it is. And they said they can make a site visit, and they can send you warning letters and seize your product if you're not compliant. But I think that this final rule, as you can see, does not resolve all the issues that are existing with herbal remedies. And so there's still, the burden of proof is still on the FDA to show that a product is unsafe. And so while we've made progress, I think I would say it's safe to say that there's a ways to go. Okay, so there's an organization called the U.S. Pharmacopoeia, which many of you may know about, and it was authorized by the U.S. Food and Drug Act of 1906. And what this nonprofit, non-governmental health organization does is it sets standards for all the drugs that are manufactured or sold in the U.S. And so as part of this, what they do is they publish something called the U.S. Pharmacopoeia National Formulary, and they make available standards for people to purchase so that they can test their product against the USP standard. And so this is really the gold standard, I would say. And they've started to include monographs and information about herbal remedies in their publication. The other thing that's important is that they also have this independent verification program. So if you as a manufacturer wanted to have your product verified by USP that it is what you say it is and it's of good quality, then you can ask them to do that, and if it meets their standard, then you get to put this nice logo on your product that says USP verified. And that really is the gold standard, I believe, for knowing that what you're taking is really what you think it is. And there's a whole list of them on this USP website if you're interested in looking at that. But if you go to the store and you see this label, then you should feel fairly confident that what you're taking is what you wanted to take. But I don't want to lay all the blame on just these problems with regulation and so forth. And I want to point out to you that it's important to know that standardization of herbal remedies is not simple. And there are a couple reasons for this. The first thing is that a lot of these remedies don't just contain a single plant. They may contain multiple plants and other additives, particularly in these traditional Chinese medicines. In addition to that, it's frequently not known what is the active component. So if you're taking something to boost immunity, what of that complex mixture of compounds within that plant extract is actually doing that? And oftentimes we don't know. So the other big issue has to do with the variability of the plant extract itself. First of all, it's very important to correctly identify the plant. And it's probably easy to mess that up. The other thing is what part of the plant is being used to actually make the product. And so the potency could vary depending on whether you use the stem or the leaf or the root. And then finally, there are all these growing conditions and processing, which is pretty obvious. But the thing about the growing conditions, I think a nice analogy is to think about wine. And for those of you who drink wine, you know that a particular vintner has grapevines that they use every year to grow grapes. And every year they make wine. And from year to year, that wine is not the same. And so it's the same problem with herbal remedies and plants, is that it's going to depend on what the conditions were, how potent the particular component is that you're interested in, and a lot of times we don't know what that is. So there's a lot of complexity to this. So the question might be, okay, it's really, really complicated, and what can we do about this? And one of the things that I think that we can do is that we can educate ourselves about these herbal remedies. And one, I think, useful source of information is the National Center for Complementary and Alternative Medicine at the NIH. And they actually have this really nice website where they talk about all the research that they fund, because that's one of their missions, is to explore CAM practices in the context of rigorous science. They train CAM researchers, and they also make available to you information about commonly used herbals. And it's a very easy, navigable website with references, and I would trust that information. So I think that that's a nice source for information. Another good place to go is SCOP, and these monographs are published in a book, which you do have to order and obtain, but they are the evidence base for clinical use of herbal medicines in Europe. And it turns out that there's been a long tradition of use of herbal remedies for standard treatment of patients in places like Germany. And so these monographs now have been available for quite some time, and the regulatory system in Europe is such that manufacturers have to meet more stringent standards than they do in the US. So this is another, I think, good resource. And I've listed a bunch of other ones here on this slide. They're, again, on that handout that I have at the back of the room, and there are a couple things that I want to point out about this list. One is that at the UW here, if you go to the UW homepage and you go to the UW Care Provider Toolkit, there are a couple of useful links that will provide useful information. And one is a page that was set up by the Department of Family Medicine, which includes 24 mini reviews on commonly used herbals, and then another one that links you to this Natural Medicines Comprehensive Database. A lot of these require subscription. You have to pay to get the information. I think that if it's something that you're really interested in, it's definitely worthwhile to do that, and I really wish that more of this was just available free to the public. Unfortunately, you have to pay for quality, is what I think the bottom line is. Okay, so now we're at the promise. And this is a picture of the Anopheles mosquito, which, as we heard about a couple weeks ago, is the insect vector for malaria. And the thing that I'd like to remind you of is that this is a huge global health problem. It causes up to 500 million new infections per year and causes up to 3 million deaths per year, mostly in children under the age of 5. But one thing about this disease that's related to the topic today is that since 2005, the World Health Organization has recommended as first-line treatment, all treatments include Artemisinin derivatives. And I think you might remember that I mentioned that Artemisinin is derived from a plant called sweet wormwood, or Qinghasu. And currently there's this very large ongoing project called the Artemisinin Project. And what their goal is to produce a consistent, cheap, and reliable source of semi-synthetic Artemisinin. And the goal of doing this would be, of course, to reduce the cost of therapy from something like $2.20 per adult course to something that's affordable for people in these endemic nations, about 10 cents per adult course. And they hope to do this in the coming years. But I think it's informative to learn a little bit about how Artemisinin came to be developed. And so what I've put here is a timeline of how things kind of came to be in terms of Artemisinin. So as early as 340 AD, it was found in the Chinese literature that Artemisia, this plant, was used in a mixture of traditional Chinese medicines to reduce fever. And this kind of went on for a very long time until 1967, when the people in the People's Republic of China decided that they were going to start a systematic search of their indigenous plants for a source of drugs. And then 4 years later, one of those scientists discovered that if they made an ethyl ether extraction of Artemisia annua, that they produced a product that produced encouraging results in Plasmodium-infected mice. And then a year later, these scientists were able to identify Artemisinin or Qinghaosu as the active ingredient. And this was testable in an animal model of Plasmodium and could be compared to existing treatments and was found to be very, very successful. And one anecdotal point that I included in here is that when they tested plants from a particular province of China, the potency was 0.5% and significantly greater than plants that were grown in the Washington, D.C. area. And then in 1979, finally they were able to test Artemisinin in patients who were infected with Plasmodium, and they found good success in malaria, chloroquine-resistant malaria, and cerebral malaria as well. So there is still a ways to go with Artemisinin. I think that what this story does is it shows you what the promise is of herbal remedies and the potential that they have for use in conditions where there are not good alternatives. So finally, I'd like to talk to you about the work that we've been doing in our lab, and that's been going on in Steve Poliak's lab in the Department of Laboratory Medicine. So first, I'd like to tell you a little bit about Silymarin. Silymarin is derived from this plant, Silymarinum, which is the milk thistle plant, and it's actually extracted from the seeds and has been used for centuries in the treatment of liver and biliary diseases. It's commonly used in Europe and Asia, and of course here it's available as a dietary supplement. So like many other plant extracts, Silymarin is a mixture of many compounds which are shown here. And in this case, they're mostly polyphenolic flavonolignands and a single flavonoid. And of course, people take this, as I told you, for hepatoprotection, for chronic liver disease. And in Europe, it's been used to treat acute liver toxicity due to poisonous mushrooms and other liver toxins. Unfortunately, it's not available in the US for these indications. So the first question that you might ask is there any clinical evidence that this stuff works in hepatitis C virus infection? And the answer is in this title. Unfortunately, the data are inconclusive. And it's a complicated slide. There's lots of information on here. But really what I wanted to point out is that the numbers of patients who have actually been studied is pretty low. And the types of Silymarin that have been used in these studies is variable, as are the dosages. And the outcome variables have even been somewhat variable, although most frequently what's been used is the ALT, or the alanine aminotransferase in plasma or serum, which is a clinical measure for liver damage. So to write this problem, to try to establish whether or not there's efficacy for Silymarin in chronic liver diseases, NCAM and the NIDDK have gotten together and sponsored a study called SYNC, which is currently ongoing, and it's a phase 2 multi-center randomized clinical trial of Silymarin for the treatment of chronic hepatitis C virus infection and NASH, which stands for non-alcoholic steatohepatitis. And it's being conducted at 4 clinical sites in the Northeast. And they are using a German preparation of Silymarin called Legalon at much higher doses than those studied in the past, although they've already done the proper phase 1 safety studies as well as pharmacokinetic studies. And they will be comparing these higher doses with placebo after treating for 24 weeks in about 153 patients who are infected with hepatitis C virus. And again, their endpoint, which is the most practical endpoint to test in terms of hepatoprotection, is actually measuring ALT reduction in these patients. So it will be a couple years probably before we know the results of whether or not there's any evidence for efficacy of this drug. And more information can be obtained at the clinicaltrials.gov website if you're interested. So the next question really is whether or not there is some rationale for Silymarin's potential efficacy against liver disease progression during Hep C infection. And in order to try to address this question, I have to tell you a little bit about what we know about the pathogenesis of liver fibrosis progression in hepatitis C. So this is greatly simplified, of course, and there is a black box here in the middle for a good reason, and that's because there are a lot of things that we don't know and are not well defined yet. But what you can see is that when hepatitis C virus infects the liver, it actually infects the liver cells or the hepatocytes, and it induces an innate immune response in those hepatocytes, which leads to the secretion of chemokines that attract adaptive immune cells. And the infiltration of T cells into infected livers is actually, in the portal areas, is actually very typical of hepatitis C virus infection. And when those adaptive T cells get to the liver, they recognize that the liver is infected by something foreign, and they then go about doing their business, which includes killing infected cells and producing pro-inflammatory cytokines. And that's here. And so this, in turn, we believe causes the death of infected hepatocytes, and when they die, they lyse and they release alanine aminotransferase, which is what we measure to measure liver injury. And in addition to that, we believe that these functions and the cytokine milieu that's generated by these reacting lymphocytes, what they do is they activate various cell types in the liver, which, as I said, was greatly simplified for this slide. But in particular, they're activating this particular type of cell called the hepatic stellate cell in the liver. And these stellate cells are resident cells that when they become activated, they're able to make lots of collagen and other extracellular matrix materials for the wound healing process. And so in the process of doing that, if this process is dysregulated and they're making too much collagen, there are too many of them, they're too activated, then it's thought that they then are contributing to fibrosis accumulation and progression of disease and eventually to the development of cirrhosis. And so you can see that if you can perturb this process or ameliorate what's going on here, that you may be able to theoretically delay or prevent the progression of liver disease. And so many groups have studied Silymarin effects both in vitro and in murine models, and they found that Silymarin actually has antioxidant effects, anti-inflammatory effects, anti-proliferative effects, anti-fibrotic effects, and immunomodulatory effects, but not really in hepatitis C virus infection. So are there any data in support of these potential mechanisms by which Silymarin could protect the liver in hepatitis C virus infection? And so we set about to try to answer that or to address that. And to do that, we obtained MK001, which is a standardized Silymarin preparation from our collaborator, David Lee, who is at Harvard. And he is a medicinal chemist who obtained this product from his collaborator in China, who verified the identity of the product, of the leaves, the seeds in the plant, and sent them to David, who then analyzed them very carefully to make sure that they were what he thought they were. And what he's done is standardize them so that the total of all the flavonoligmans together compose greater than 90% of the material, and that every component is completely characterized and identified. So this is the product that we've used for our studies. And the first thing that we did was we tested them to make sure that there was no toxicity that was associated with using these in PBMCs. And I didn't have time to include that data, so I won't show it to you. But in the concentrations that we're using for these experiments, there was no toxicity to the PBMC. So in this first experiment, we wanted to look at pro-inflammatory cytokine secretion by peripheral blood mononuclear cells that were obtained from a hepatitis C infected person. And so what's shown here are TNF secretion and interferon gamma secretion by peripheral blood mononuclear cells. And what you can see is that in the absence of any other type of stimulation, there's no effect of MK001 on either TNF secretion or interferon gamma secretion as detected by ELISA in culture supernatants. But if you stimulate T cells with plate-bound anti-CD3, alone, then you see that the PBMCs are making TNF and interferon gamma, shown in these gray bars. And with increasing amounts of Silymarin, increasing concentrations of Silymarin compared to the solvent control, shown in white, and the MK001 treated cultures in the colored bars, you can see that there was a dose-dependent effect on pro-inflammatory cytokine secretion using MK001. So then we wanted to know, well, what about the T cells specifically, because this was a heterogeneous mixture of peripheral blood mononuclear cells. And so what we did was we immunobead purified them so that they were quite a pure population. And then we tested those T cells specifically in culture again, looking at TNF-alpha, interferon gamma, and IL-2 secretion. And in all cases, you can see that by comparing treatment with solvent versus treatment with MK001, that there was a substantial inhibitory effect on the secretion of pro-inflammatory cytokines. And we decided to look in yet another different way at this case. And so we used flow cytometry so that we could specifically identify both CD8 positive T cells on the y-axis here in the upper quadrants, and the presence of cytokine detected intracellularly, TNF in the top panels and interferon gamma in the bottom panels. And so what you can see in this upper right panel is that with anti-CD3 stimulation that you saw a substantial TNF secretion by CD8 T cells and also interferon gamma secretion. And that when you added MK001 at the time of stimulation, then you were able to inhibit the amount of TNF and interferon gamma that was present in those cells. And we also looked in the case of Jurkat T cells, which is a transformed T cell line. And because these cells are transformed, they are proliferating spontaneously. And similar to data that I didn't really have time to show you in primary T cells, what we found is that treatment with MK001 could inhibit proliferation as detected using a standard tritiated thymidine incorporation assay. And that, again, that treatment with MK001 inhibited IL-2 secretion when cells were stimulated with anti-CD3 and anti-CD28. So it seems to have a very consistent effect. We've tested people who are infected, people who are uninfected for these effects on pro-inflammatory cytokines and really find a very consistent result. So we know that proliferation and pro-inflammatory cytokine secretion can be controlled by a transcription factor called NF-kappa B. And so what we asked in this experiment, which was performed in collaboration with Steve Poliak's group and Keith Jerome's group, is whether or not MK001 inhibited NF-kappa B transcription in these Jurkat T cells. And so what we did, we took the Jurkat T cells and transfected them with a plasmid that contained a luciferase reporter gene under the control of an NF-kappa B dependent promoter. And then we stimulated these cells with either TNF, anti-CD3 or anti-CD3 plus anti-CD28. And what you can see is that compared to the control, that there was a dose-dependent effect in all cases of inhibition of NF-kappa B dependent transcription in all those cases. And because we know that NF-kappa B also controls the expression of other genes that are involved, for instance, in this case, in cell-cell interaction, which is ICAM1 is an adhesion molecule that's very important in cell-cell contact and in situations like with antigen presentation and also with killing of infected cells. We looked to see whether MK001 could inhibit this, and we found that in fact it did. So what the green and red lines show you are the expression levels of ICAM on peripheral blood or CD3 positive T cells, actually, that were stimulated with PHA. And this is in comparison to this gray region, which are unstimulated cells. But when you add MK001 at the time of stimulation, then you see this inhibition of ICAM1 upregulation. So then knowing this data, I hope that you believe that MK001 really has these effects on the immune response. But you might say, wait, if you inhibit the immune response in a viral infection, then what's going to happen to the virus? Isn't the virus just going to go crazy? And then how does that really help anything? And so Steve's group had actually published these, the results of these experiments earlier, which were actually quite surprising. And that is, what he did was he took human hepatoma cells in culture and treated them with MK001 at varying doses, shown here. And then he performed this Western blot to look at expression of HCV proteins after JFH1, which is a type of hepatitis C virus that can infect T cell or hepatoma cells in culture. And what he found was that compared to this control, which is the solvent, that increasing amounts of MK001 actually inhibited viral replication, as was true for treatment with interferon alpha. And then he also looked in a therapeutic situation where he first infected cells with either JFH1 or did a mock infection. And he could show that treatment with interferon or with MK001 resulted in much less expression, much diminished expression of these hepatitis C virus proteins. So these studies indicate that MK001 not only has effects on the immune response, but they also has effects on the virus. And then more recently, a paper was published led by a group led by Peter Ferenczi. And what they did was they actually treated people with Silymarin. And Silymarin is a new term that I haven't introduced before. It's actually a component of Silymarin. And they gave people who had previously not been responsive to pegylated interferon and ribavirin very large doses of Silymarin intravenously. And they treated them this way for 7 days. And after 7 days, then they added pegylated interferon and ribavirin. And as you can see, that over time, these viral levels in people continue to decrease. And so there was a dose-dependent effect of this IV Silymarin on viral levels in humans. So this basically confirmed the data that, that Steve had earlier published in gastroenterology. So what do we think is really going on overall, and how could Silymarin be working? Well, this is our model. And here we see hepatitis C virus infects hepatocytes and causes problems like increased reactive oxygen species and NF-kappa B activation, which leads to secretion of inflammatory cytokines and chemokines. And as a result of the inflammation that's occurring, we see T cells homing to the liver and T cell receptor engagement, NF-kappa B activation and proliferation, pro-inflammatory cytokine secretion and killing of infected hepatocytes by T cells, all of which together are contributing to liver disease and activation of stellate cells and extracellular matrix deposition, as I discussed before, in addition to liver damage. And so what we think is happening with the addition of Silymarin is that Silymarin is actually acting at multiple places to inhibit viral replication as well as to inhibit immune responses, and thereby sort of dampening this whole process that contributes to liver disease progression. And one thing that I didn't mention is that we definitely have data from Champapal when she was in the laboratory that showed that there were also antioxidant effects that were occurring in the system as well. So I'd like to recognize the contributions of all of our collaborators, including the people in my group who've done all the immunological experiments, Cha Wang and Maggie Schuhart, who have recruited all the patients that provided the critical material for us to test, Steve Poliak's group, Keith Jerome's group, who helped with the NF-kappa B experiments, David Lee, who provided the Silymarin, and Steve DeRosa and Brent Wood, who've provided critical and helpful advice, technical advice for our experiments. So overall, what I'd like to conclude is, and what I'd like you to take away from this talk really, is that herbal use is very, very common and widely accepted by the general public. They're complex mixtures that are difficult to standardize and to regulate, and education and access to quality information are really critical to ensuring the safe use of these herbal remedies. But we have to remember that herbal remedies have great therapeutic promise and that much more research is needed in this area. So as a public service announcement, I like to say that, if you're going to take an herbal remedy, read the label, but it might not be true what it says on the label. Educate yourself. It might not be easy to find out all the information that you want, and be an activist. If you're a healthcare provider, ask your patients specifically about herbal remedies, what they're taking, and educate them. Encourage them to report adverse effects. Educate students, residents, fellows, and colleagues, and advocate for better legislation and more funding for regulation and research. So that's it.
Questions? Yeah, Brad.
Interesting talk. I wonder if you could tell us, does Silymarin have direct antiviral activities, or is it decreasing viral replication because hepatitis C replication requires some level of post-cell activation, which is interrupted by Silymarin?
We think, and Steve can speak better to this than I can because he's done these experiments, but we believe that there are direct effects on the virus and does not rely on activation of cells. Jeff?
Some complementary medicines are given in sort of what are called homeopathic doses, meaning that it's can be diluted incredibly far to the point where it's difficult to identify that it's there anymore. Are those studies funded by the NCAM, or are they even considered to be ethical to do a controlled trial with those? I don't know what placebo would be, they're almost hard placebo, but.
Now, I have actually gone through and looked at the list of funded studies by NCAM, but I obviously don't have access to everything that they've ever funded. It certainly falls under their realm of things that they study. I mean, it's definitely not just limited to herbal remedies. It's not, so CAM is a very broad term, that refers to acupuncture, mind, body, health, all those kinds of things that are difficult, prayer, things like that are very difficult to quantify. And so that type of research is difficult. I think so the answer to your question is yes, they probably do fund that. I don't know what kinds of things are active. But even in those cases, there have been reports where people say, well, there's so little of it there that there's no way that it could hurt you. And in fact, people have detected bad things in those preparations.
Go ahead.
Regarding Artemisinin, it works for malaria. What other things have they looked at, and specifically have they used it for certain cancer therapies? Are you aware of any of this?
I'm sorry, I can't answer that question of whether or not artemisinin is useful for other things. I seem to recall that it may be, but I can't really give you any specifics, sorry. Dr. Weiner.
Thank you for a beautiful combination of public service and science. Wanted to know whether you think there's something particular about the immunosuppression from the Silymarin-related drugs that would make it more favorable and less likely to have other consequences from immunosuppression. Is there reason to think that it's more targeted, or obviously it has the double action of being an antiviral as well as an immunosuppressant, but are there some advantages that you think might be possible as an immunosuppressant?
Well, so there are a lot of things about Silymarin that I didn't have time to tell you, but you bring up a really good point, and I think the issue that you may be getting to is this whole issue about generalized immunosuppression and how that might affect somebody who's infected. And so one of the things about Silymarin that we know is that it's got a very short half-life, and it's metabolized in the liver fairly quickly. So peak levels occur at about 2 hours, and then the highest it's partly excreted in the bile and partly in feces. And so the highest levels you would expect to see would be in the liver. So that's one thing about maybe how it's sort of specifically targeting what's happening in the liver. And you might not see those effects very easily in the periphery. And so no one knows, of course, what happens when you're chronically taking this, even at lower doses for a very long period of time. But I think that one sort of hypothesis that is certainly unproven that I have about its immune effects is that if you are talking about the immune response as kind of an initiator, playing a role in kind of setting off the inflammation and kind of getting things rolling and kind of keeping things going in a way that's undesirable for the overall health of that organ, that if you, and we know that hepatitis C specific responses are actually attenuated because probably because of the large number of Tregs that are around to control those responses, that if Silymarin has this sort of mild effect on these Hep C specific responses, then it might be able to specifically affect those responses given its short, sort of the short-acting nature of it and the targeted sort of, targeting of it to the organ that might specifically help prevent sort of deleterious immune responses that you don't want, but maybe not keep you from making the kind of response that you need if you get some kind of massive, some kind of big bacterial infection that you really need to fight off. So there really isn't any, as far as I'm aware, reports that people who take Silymarin are somehow immunosuppressed or anything like that.
I want to thank Dr. Morishima. If you have additional questions, maybe she'll stick around.